On behalf of the University of Montana Alumni Association and the Community Lecture Series Committee, it is my honor and pleasure to welcome you to the 2023 Community Lecture Series. We're going up against Grizzly basketball tonight, so I think that's where some of our attendees are. This series, now in its 25th year, showcases some of the outstanding faculty here at the university and the work they do in advancing research and learning. We'll begin with the university's statement of acknowledgement. The University of Montana acknowledges that we are in the Aboriginal territories of the Salish and Kootenai people, or Kalispel people. Today, we honor the path they have always shown us in caring for this place for the generations to come. Now that we are back to a live in-person lecture format, we are trying something new with respect to ticketing. Rather than list a price for the ticket, we invite you who are enjoying these lectures to pay what you think it's worth. We've suggested $30 for the whole series or $10 for individual lectures, but we hope you will determine the value yourself of what these lectures are for you. It's a risk, we know, but we are confident you can figure out for yourselves what you are gaining in learning and enjoyment. And as always, if you know of someone who would enjoy these lectures here with us or over Zoom, please feel free to spread the word. If you haven't already registered, please do so through the University of Montana Alumni Association webpage. These lectures are being video recorded by our friends at MCAT, Missoula Community Media Resource, for later cablecast and posting to YouTube. Later, during our Q&A, we will be handing around microphones to capture questions and comments from the audience for those watching over the internet and for later cablecast. Please take a moment at this time to silence your personal devices. The seal of the University of Montana features a hand holding high a torch and bears the university's motto, Lux et Veritas, light and truth. Our university is a place where students come to study, learn, and conduct research in an effort to pursue light and truth for themselves and for others. The university's efforts at supercharging this effort was recognized this past year when it was awarded R1 designation as one of the nation's top research institutions. It would be daunting to provide you with a lecture series that touched upon all the areas in which the University of Montana faculty are doing research that earned that award. But we are happy to present a series where you can gain some appreciation of the world-class research that is going on across the campus. The committee has selected Dr. Mike Minnick as our third lecturer in this year's series. Dr. Minnick's academic background includes 2 degrees in biology from Walla Walla University and a Ph.D. in microbiology from Washington State University. In our world, there are terrors, great and small, and Mike Minnick's time is spent among the very smallest terrors as his research leads to ways to protect us from them. Please join me in welcoming Dr. Minnick as he presents his lecture, From South America's Mountains to Montana, Revealing the Secrets of a Neglected Bacterial Killer. Please welcome Dr. Mike Minnick.
All right. First, I want to thank Jeff for a kind introduction and for the CLS committee for the invitation to talk with you tonight. And I would like to thank you for spending some time with me tonight to share with you some of the research my lab has been doing for the last 32 years at the University of Montana and spending your tax dollars. So my talk will be consisting of ways of knowing, the subject category of this series, about the history and biology of Bartonella bacilliformis, one of the bacteria that we work with in my laboratory. And we're going to describe how this bacterium colonizes humans, as well as strategies used by this bacterium to invade human tissues. And then we're going to flip the coin and look to see how this bacterium interacts with a sandfly vector that is used in its transmission. And then finally describe some research that we've done on how this pathogen persists in both humans and insects, 2 very different backgrounds. The history of Bartonella bacilliformis, the agent that we're going to talk about tonight, is very rich. And since pre-Columbian times, the Incan artwork, both ceramics and huacos figurines, have depicted lesions from this particular infection called verruga peruana. Verruga peruana refers to blood-filled warts that occur on the skin during the infectious process. More recently, the first description of these verruga lesions actually occurred in journals of the conquistadors as they were conquering South America, and this was roughly in the 16th century. The first published treatise for this particular disease occurred in the 17th century, and Pedro Gago de Vadillo, my Spanish is a little rusty, but he did this treatise on the disease. And finally, in the late 19th century, we had roughly 6,000 workers that died of a mysterious fever illness while constructing the railroad between Lima and La Oroya. And so to this day, we use the term Oroya to describe the fever caused by this particular bacterium. Now let's back up. This particular disease caused by Bartonella bacilliformis is referred to as Carrion's disease. It is named after a medical student, Daniel Alcides Carrion, and he was a medical student in Lima, Peru in the late 1800s. And at this time, the germ theory of disease was just coming into vogue with Pasteur and Koch, who suggested that disease was actually caused by some etiologic agent. And being a bright young man, he noticed that in his travels throughout Peru, there were people having Oroya fever as well as these verruga peruana lesions, as you see here in this lower left-hand diagram, essentially a blood-filled wart. And he reasoned that somehow these two diseases were related to one another. And he hypothesized this and did some self-experimentation. Now, I've given a lot to my research in terms of sweat and blood, but this guy gave it all. And so he injected himself with the lesions from a verruga peruana patient. And within days, he developed symptoms of fever and muscle pains as well as anemia. And he carefully took notes. About a week later, he was too sick to even take notes, and his colleagues had to resume his activities at the med school. And then in October of that year, 1885, he died of Oroya fever. But he definitely demonstrated that the two syndromes, the skin lesions as well as the Oroya fever, were one and the same disease in a very different presentation. Now, to this day, if you go to Peru, and I've been lucky enough to do this, his name is everywhere. And he is found on postage stamps, hospitals, license plates, statues, institutes, and on and on. We don't even know his name in North America. In about 1909, during the Peruvian expedition, 1913, the name Bartonella bacilliformis was coined by Richard Strong, who led that expedition, based upon the work of Alberto Barton, who described the bacterium and had his name forever enshrined in the genus of this bacterium. Microbiologists are weird. That's really a great honor. So what is Carrion's disease, and how does it present in humans? This is a strange illness for the reasons that I just spoke about, in that we have two phases. We have an acute disease that has a fatality rate of up to 88%. It makes the plague look like a picnic. This is one serious illness. And of course, rates like this were common before antibiotic therapy was available. And during 1 to 4 weeks after the infection starts, you have about 80% of the red blood cells that will become lysed. And of course, as a result of this, you have tremendous anemia. Along with this anemia, we have immune suppression. We have secondary infections like salmonella that takes advantage of this situation. We have muscle pain, pallor, as well as jaundice. If you look at a blood smear from a patient with Arroyo fever, virtually every red blood cell is infected. So the red cells are the large spherical objects there, and each of those little purple specks is a bacterium. And I defy you to try and find a red cell there that doesn't have a bacterium. If you do a scanning electron micrograph and you get some ultrastructure, you can see the bacterium invading red blood cells and deforming the surface of the membrane to make little pits where it can enter. This particular disease is very nasty for children, pregnant women, and their unborn child, as well as travelers who visit this region, and it's a complete new experience in terms of their immune system. On the flip side of this, we have a second part of this biphasic disease called verruga peruana. And so these were the lesions that were found on the Incan ceramics, and here we have a variety of examples for you. This is a picture I actually took in Peru of a child's leg. But this secondary stage of this infection is chronic. It can be a lifelong infection for these patients. And these blood-filled warts are called verruga peruana, as I've already mentioned. It is rarely fatal at this stage, and it is accompanied by scarring and persistent blood infection. So as a result of this persistent infection, we have a carrier state, and that person can serve as a reservoir for this bacterial disease. So inhabitants in endemic regions, the Andes of Peru, Colombia, and Ecuador, are at very high risk for this illness. The Peruvian government, before the recent meltdown, was kind enough to invite several Bartonella scientists to visit the country to help them develop a vaccine in 2008. I was lucky enough to do this. And after driving and driving and driving, we were finally in the Andes region above Casma, Peru, and the locals were extremely generous of their time. We were offered meals. I had the choice between fried guinea pig and chicken, and I took the chicken's way out. I just couldn't go for the cuy, as they call it. But what we found was absolutely sobering, and it inspired us to work even harder on this illness. Literally, every other child had verruga peruana on their extremities. And it was heartbreaking to see the incidence of disease. Now, granted, this is anecdotal, and we weren't taking statistics, specs, or anything like that, but the disease was everywhere. The historical range of this disease, as I mentioned, it is found in Colombia, Ecuador, and Peru. And it's found at very high altitudes, although for a Montana audience, this isn't particularly high, 1640 feet. We're almost twice that much here in Missoula. But it can, in fact, go up to 9,800 feet, and the Arroyo Railroad was at about 12,000 feet. And we think that this reflects the fact that the sandfly vectors for this illness live at those high altitudes. And the two major species of sandflies that are responsible are Lutzamia vericarum and Lutzamia peruensis. And here we have the male and female of vericarum. Only the female takes a blood meal, almost like a mosquito. And she needs to take the blood meal in order to produce eggs, again, like a mosquito. What we have noticed over the last 2 years is something very frightening from a public health perspective. We've had the range of this once restricted pathogen extending into lower areas, including the jungle of the Amazon, at least the high Amazon, as well as coastal regions, which are much lower than the Andes, of course. And we think what is happening is that there are additional sandfly vectors that are coming into vogue or being employed by this pathogen. Climate change undoubtedly has increased the range, the habitat of this organism, and perhaps other Bartonella species are in fact giving us symptoms of Verruga peruana. And just published a paper on this today. It just came out. But here is an estimate of the disease incidence. 1.7 million people are at risk in an area roughly 56,000 square miles. So it's a significant chunk of South America that is at risk for infection. Now, the thing that has really driven my research and my ways of knowing is that by the World Health Organization standards, this disease could be eradicated. We have a human reservoir, so we could eradicate it by virtue of a vaccine if we had one. The organism is very restricted in terms of its localization, and we have a pretty good idea of how the disease is transmitted between sandflies and humans and back again to sandflies. So with that vaccine, potentially we could stop this disease once and for all. Hopefully I'll be retired. But Bartonella bacilliformis, what is it? Here is an electron micrograph of the bacterium. Looks like a hot dog with lots of propellers. Those are the flagellae that allows it to move. And this particular bacterium, as well as his cousins, are bacterial vampires. They live inside red blood cells. This is a very unusual place for any bacterium to live. And mostly when you think of this, you think of parasites and not bacteria. This is a hemotrophic organism because of that. It is a gram-negative bacterium, which means it looks structurally more like the E. coli in your gut than the staph that lives on your skin. And we'll show you a picture of this in a little bit. With those swimmers, he is highly motile. He can move like nobody's business. And when you look at a family tree, this is all the Bartonella that are grouping down here in these lower branches, but some of the closer relatives you know. Brucella is a cousin of Bartonella, and of course that infects elk herds as well as bison and lives up here in North America. A little bit of clinical aspects of the disease. Diagnosis is done by blood smears. Very simplistic, on-the-spot way to diagnose a disease. It is very inexpensive, has high specificity, but low sensitivity. And we're trying to develop some new antibody and PCR tests to help. But even if we get these tests, having it in these rural villages is going to be a big ask. How are we going to get them trained? How are we going to get them the expensive equipment and so forth? If somebody does have Oroya fever or Verruga peruana, we can treat them effectively with antibiotics. But again, if you're living at 12,000 feet in a small village, you don't have good access to health care, let alone antibiotics. And prevention of this disease could be simply done by education, use of insecticides, avoiding sandflies about dusk, and using bed nets and window screens in the homes. They don't have doors, they don't have window screens, they don't have flooring. It's very primitive, and sandflies love it. So how does this bacterium colonize human tissue? We think that motility, the ability to move, is very important for these bacteria. When the female sandfly takes a blood meal and injects the bacterium into that circulatory system, those bugs can passively move to organs that have lots of circulation in them, like the liver and the spleen. So that's a passive way. A second is by that motility, by virtue of the propellers, the flagellae that I showed you earlier. One of the things that we've noticed for some time, and certainly the skin lesions bear this out, is it has a tropism for the skin, and we think he likes things a little bit on the cool side, and that's why this is the case. Finally, that motility, especially of the flagellae, could increase interactions with host cells, much like a sperm and an egg. Good swimmer, binding to a host cell, enhancing infection. So one of the first things that we did in my lab, and this would have been about 1993 with my first graduate student, and he just knocked on my door one day and said, "I'm here." And now he's a patent lawyer, so he makes good money, and he's putting his biology to good use. But the first question we asked was, does the immune system see these swimmers, and are they involved in host cell attachment and invasion? And so we politely asked a rabbit to make some antibodies for us, and we did a crude preparation of these flagellae by simply shaking the cell and then purifying those out. And what we found is that antibodies could indeed recognize these appendages, and each of the little subunits that you see here in this propeller are represented by this arrow right there on this gel that we have purified and separated based in an electric field. So the question remained, does motility help these bacteria parasitize red blood cells? Is it helping them get in or to touch them? And in order to answer this, we said, well, we've got an antibody to that propeller. What happens if we add it to these bugs and ask, how do they interact with human red blood cells? And so here you can see that anti-flagellin antibodies that we showed up here can actually, compared to two different controls, significantly reduce the amount of association with these red blood cells. But more importantly, those same antibodies can almost completely stop any invasion of those red blood cells. So this could be potentially a vaccine candidate for stopping this bacterial parasite, because it certainly does wonders at stopping that invasion. How does Bartonella invade cells? Well, Bartonella tends to have two favorite cells on the menu. The first one are those red blood cells. And we're not going to go through all of these different strategies that are taken by these bacteria, but you can really see this red blood cell. This is a human red cell with a blue-colored bacteria. It is deformed. And again, these bacteria have the amazing ability to deform the surface of these cells to enhance uptake into the cell. The second favorite cell type for parasitizing are the cells that line the circulatory system, the endothelial cells. We also find these in the lymph tissues. Another graduate student, and Sam is now, he's an MD, Ph.D., and he heads the health clinic at Montana State University, got his Ph.D. with me years ago. And we asked, if we were to shotgun clone, so just randomly take pieces of the DNA from Bartonella bacilliformis, clone that and stick it into E. coli, which we all have in our gut, will it make it an invasive bacterium? And lo and behold, on the first time out, Lucky Sam was able to show that, sure enough, we were able to have human red blood cells, shown here, it looks like a big Pac-Man, with E. coli that would normally not do this activity to invade these red blood cells. And we called the gene that was responsible for this the invasion-associated locus B. And we also did some mining at that time of genes that were nearby and published on those. But now, with whole genome sequencing happening around the clock, around the world every day, we can get a good feeling for what this gene is doing. So we asked, and this was some of Sherry Coleman's work, another PhD student, we asked, what would happen if we were to knock out that invasion-associated gene in Bartonella? Would it lose its invasiveness? And here you can see a parent strain of the bacterium, and here is a mutant, and we have significant reduction in its invasiveness. And as molecular biologists, we have to say, well, all right, that's half the story. What if we put that gene back in that strain? Will it bring that invasiveness back? And so here we have the parent strain, and here we have the gene brought back in, and sure enough, it becomes invasive. So the invasion-associated locust B gene has been found in not only Bartonella and other Bartonella species, but also Brucella, its cousin. One of the tricks we use as microbiology in order to understand what these gene products are doing is to strip away the cell like an onion. So we get different pieces of the onion, and we ask, where is that protein found? And the IOB protein, if we use our antibody, we find it in the inner membrane. We were surprised. We were thinking it might be in the outer membrane where it could talk to host cells, but it actually ended up being in the inner membrane. And in the mutant, we don't have any of that protein, as you would expect. And then when we brought the gene back, we get the protein reestablished and produced. One of the coolest stories we found about this invasion locus is that the expression of this particular gene depends upon environmental cues. So we get the most RNA, which encodes for the protein, when it is acidic. And we get also a high level of that RNA at lower temperatures. You can also see that the protein follows suit. At acidic temperature, we get more of the ILB protein, and at cooler temperatures. So we scratched our head and said, well, what the heck is going on here? But if you look at the biology of this bug, I shouldn't use that because we're talking about two bugs here, but we have mama sandfly, and when she takes a blood meal, the gut pH rises from 7 to 7.4, which is the pH of our blood. And slowly, the temperature of that blood is going to fall from 37 to 20 degrees. And she will then digest that blood over the next 2 to 3 days, and the gut will acidify. She will then take another blood meal in about 4 to 5 days, and during this time, we have an acidic condition as well as a cooler condition that is experienced by this bacterium. So our hypothesis at this time, or our working model, is that in the gut, the bacterium becomes primed to reinfect the next round of human red cells as that sandfly takes a blood meal, because there are high levels of that invasion protein. Whereas in humans, we have downregulation of that, and this, in fact, would prevent red blood cells from continuing to be infected, and basically it helps in the eviction from the bloodstream into those endothelial cells that lie in the vasculature. So what does this protein look like? Well, we still don't know how it works, even after all these years and all the different labs that have worked on it. But here you can see in this bacterium, we've got 2 layers in the cell wall of membranes. And here is the invasion locust B protein. And I'm hedging my bets here because our work showed it was in this membrane. Other Bartonella have it in the outer membrane. So I've kind of drawn it right between the two. So I'll compromise. So what about that sandfly? If we flip this table and now focus on the vector of transmission, what is happening? And here are 2 different sandflies that we've worked with in my laboratory. This is a Lutzemia vericarum, is a known vector for Bartonella, bacilliformis. Lutzemia longipalpus is an insect model that we have used and you can buy commercially. So sandflies are the Rodney Dangerfield of the arthropod-borne disease world. They get no respect, all right? I've even had NIH officers say, why are you working with sandflies? Well, it's because that's what transmits my bug. I like them. They're actually kind of cute. I know that's weird, but. So sandflies are vectors of sandfly fever. Go to the Mediterranean, come back with more than you bargained for. Leishmania infection, which is a protozoa, and of course our current topic, Carrion's disease, which is bacterial. Here's a little bit of anatomy on the exterior and the interior. I just want you to focus or remember, put this in your back pocket, the abdominal midgut. So this is a blood meal right in there. That's why it's dark red. And we're going to talk more about that particular organ in a second. So if you go in Peru, and some of our collaborators have done these long-term studies, and I mean years, and they have done sandfly trapping to see when sandflies are active, when they are most dense in their populations, to try and get a handle on this disease and its transmission. And what we find is that the highest sandfly populations occur in, not surprisingly, the onset of the summer months, October, November, and December, and those bars are the sandfly numbers. We have the temperatures at both the AM in blue and PM in pink. And again, the highest amount of sandflies are when the temperature is warmest. Likewise, we have the highest amount when the humidity is high, not surprisingly. And these would occur in the same months. Lowest populations of the sandflies happen in the winter, which is opposite from yours and mine, June and July. This is a little sandfly trap. There's a light to attract the sandflies, and some unlucky grad student gets to collect those each day and count the different species. Transmission of Bartonella bacilliformis shows a little bit of lag time. So even though we have the highest amount of sandflies during the early part of summer, there's about a 3 or 4 month period before we really start to see transmission cook. And this is because these flies are older, and the older female flies are very aggressive, they're hungry, they want to lay eggs, and they can really pack a wallop on their bite. So they will have the highest rate of success at doing so. We also see that the sandfly activity varies depending upon the time of year. And in the winter time, just like Montanans, most of the activity is on the inside of that little house. And in the summertime, when the nighttime temperatures are warmer, we have both inside and outside activity, and it can occur throughout the night. In the winter, beginning of the evening, and that's about it. To give you an example of what these people are facing with respect to sandflies, this would be a typical village, and the homes in these villages have roofs that may be made of pieces of wood, they may be pieces of metal that are scrap, and if you were to get closer, you would see that the floors are dirt. We've got a fertilizer bag covering that window, so the sandflies can come in. They can lay their eggs, and the larvae can hatch in the floor. And frighteningly, I don't know if you can see this, but there are cracks in this plaster wall, and we could take and suck adult sandflies right out of those cracks inside the homes. So every night, these people were being bitten and potentially having this disease transmitted. So back to this climate change and the potential role for this, we notice that outbreaks of this disease are cyclical. So we can see that about every 4 years, the incidence of transmission shoots up. And this correlates very well with El Nino events, which is where nice, warm, thermal energy from the South Pacific comes into the South Pacific region. So we get rainfall, we get humidity, we get nice warm temperatures at night, and the sandfly populations explode. These are some bites on yours truly. Sandflies are crappy biters. They will bite and bite and bite, unlike a mosquito, which is an elegant insect for biting. These guys fly poorly, they bite aggressively, and they don't stop till they're full. They'll keep biting. All right, let's see if we can get this to work. So this is how we work on sandflies in my lab. We have sandfly boxes, as you see here. Here is a sandfly larval pot. And we grow the larvae in a mixture of fermented rabbit feces and Purina rabbit chow. And the larvae love it. As they are maturing, they will release, and then they will quickly adhere to that white background. Ticks like white for whatever reason, too. So we can take these sandflies, and we can suck them into our little aspirator tube, and we can then manipulate them. We can ask them, will you please become infected, and where do these bugs end up in the context of that insect? And to do that, we have mosquito feeders, which are glass-blown, kind of like a bell-shaped jar, and you can add the blood meal spiked with your bacteria, and you can maintain the temperature of that blood with a constant water flow, and you can have your little hungry sandflies, see those little black specks down there, ready to take that blood meal. But here we have the insects down here, and we've got a little piece of chick skin. You can buy frozen chicks. This is not a very fun process. You lose a lot of text by having them do this. But anyhow, spread that chick skin across there, blood meal up here spiked with the bug, and we watch to see who feeds and where these bacteria end up. Here is an example of Bartonella bacilliformis 24 hours after taking a blood meal. We have a nice way to track these bacteria. They are expressing a protein that makes them glow under ultraviolet light. Everyone likes glow in the dark, right? And each of these little specks is a bacterium, and it is found in the gut of one of these sandflies. So 24 hours after taking a blood meal, we can actually take a look at these bacteria. Not only are they alive, but they're swimming like crazy. They're happy as clams. They've evolved to live within the context of that gut. So using this approach, we've been able to study the relationship between the bacterium and that insect vector. And we find that most of the infection of the insect occurs in its tummy area. As I mentioned earlier, and I wanted you to remember, that abdominal midgut, or AM as we call it, is the primary focus of infection. The valve towards the exterior is also somewhat involved, but most of the lion's share of that infection is in that tummy. If you look at the eggs, the feces, the mouth parts, nothing. You don't see any bugs in there. And at this juncture, we think that for transmission to occur from a sandfly to a human, there's a little regurgitation going on. That it's a sloppy procedure, and they're sloppy feeders to begin with, and this regurgitation will enhance transmission of the bug to humans. This is similar to what happens during plague, as Yersinia is transmitted from fleas to humans or to other vertebrates. So the real conundrum here is, how does a bacterium live in a human and an insect and do so successfully and keep this cycling going? Before we talk about that, I just want to emphasize that this disease is very important for the people of Peru, especially. 60% of the people in endemic regions have antibodies. This is way higher than AIDS in the United States and elsewhere. 15% of these people are active carriers, live bacteria in their blood. It's a tremendous health burden, and yet no one really cares. So how in the world does this bacterium establish this long-term chronic infection? And what are the attributes that allow this to happen? And this question that I keep coming with, how does a human pathogen survive in an insect and go back to a human and repeat this cycling? Well, first of all, got to have a little bacterial anatomy. Now, way back in '94, I'm dating myself. I took a look at the LPS of this bacterium. And now we have to describe what LPS is. So if you look at the cell structure of gram-negative bugs, we've got 2 membrane systems. And the way these bacteria talk to and interact with their environment is through a very important molecule called LPS, or lipopolysaccharide. And so we've got all these little tiny chains of sugars that extend to the exterior. And they can bind things, and they can trigger the immune system to go haywire and so forth. And I found that in Bartonella, they're stubby. They're just boring. I'm like, what's going on here? So they don't have these long chain sugars. Salmonella has beautiful long chain sugars and teases our immune system to no end. Bartonella takes the opposite tack and says, I'm going to shorten this and fly under the radar. Furthermore, the LPS molecule, that yellow box and those long chains, look at the difference in toxicity between that and Salmonella's LPS. It's 1/1000th as toxic. And finally, there are molecules that literally shut the immune system down. And because of this combination of attributes, these bugs can fly under the immunological radar and keep that chronic infection going. They can evade the immune system. So here we've got the skin lesions, the verruga peruana in a young child. I did not take this picture. This is from the literature. What is this? And essentially, this blood-filled wart is production, hyperproduction of new blood vessels. Now, formation of blood vessels is very important for development, for tumor formation, and on and on. Here, it is not good. It is a pathological condition, for sure. How does it occur? It's unchecked, unregulated growth. We have inhibition of cell death that would normally occur to stop this. And our work recently implicated a growth factor called EGF, and we're going to talk a little bit about this. But before we go there, we have to kind of look at the bacterial perspective. What is in it for this bug? Why in the hell do we see this? No other bacteria on the planet does this during an infection. I think that it's creating a little bacterial nest that allows for leakage, slow leakage of this bug into the circulatory system and maintaining this chronic infection. And it may even protect against the immune system, and maybe it's used in iron acquisition. These guys love iron. So we discovered a couple years ago that if you were to infect the cells that line the circulatory system, the vascular endothelial cells, so here's a little tiny capillary, and the endothelial cells would be this pink lining right there. And you can culture these, you can buy these cells, and they love to talk to each other. They're used to talking to make these vessels. And if you infect these cells with Bartonella, boom, the amount of epidermal growth factor shoots up compared to uninfected cells. Now we have to back up a little bit and ask, well, what is epidermal growth factor, EGF? This was the first growth factor discovered from humans, and in 1962 by Stanley Cohen, and he won the Nobel Prize. I will not win any Nobel Prize in my career. But it's a very tiny protein in all kinds of cell types found throughout the body. And when it binds to a receptor found on all kinds of cells, it makes them proliferate, makes them create new blood vessels, stops them from dying, and increases survival. Sound familiar? That's exactly what is happening to make those verruga peruana. So with this discovery, we were so excited because this might be something to hang our hat on. So we asked, do different strains of this bug, this bacterium, trigger this event. And so if you have 3 different strains of the bacteria compared to an uninfected control, they all produce this epidermal growth factor, and you can measure that using various assays. We also wanted to know, does this require an infection? Does it have to have a live bacterium? And so if you treat your bacterium with formaldehyde, nasty stuff, right, carcinogen, it will cross-link the cell, the bacteria, to hell and back. But in this assay, you see that we lose all of the production of that growth factor. So we've got to have a live bug, we've got to have an active infection for this to occur. If we step back just a little bit and we ask, what happens if we block that receptor with a specific competitive inhibitor, shown there in blue? We have, again, our assays looking at proliferation, and boom, all of those that are blocked, regardless of strain or in a positive control, are going to, in fact, cause inhibition of that proliferative event. Something else that happens during production of blood vessels is that the cells that line the blood vessels move. They migrate towards stimuli. And so if you look at what's called a Transwell assay, we can actually have a little tiny membrane separating these endothelial cells, shown there as little purple spheres, and our bacteria down here, and ask, do they migrate towards that stimulus? Do they migrate towards the bug? And sure enough, by doing this assay, we see that in our positive control, as well as these 3 different strains of Bartonella, they move towards that stimulation. And if we kill the Bartonella, it loses a significant portion of that stimulation. And finally, the last thing we looked at is that these cells are progenitor cells for making blood vessels, aren't they? So it stands to reason that they would make tubes. And so here you see just standard endothelial cells in culture. They're scattered to the wind. If you add a positive control, you can actually see them talking to one another and making tubes, or precursors to blood vessels. And if you use Bartonella bacilliformis during an infection, voila, we see those same tubes starting to form. So with this migration, tube formation, and the ability to trigger division, we are forming these blood-filled warts in the skin of these children and adults that are infected. This last panel right here, number 4, shows the tube formation by dead bacteria. So in this assay, it didn't matter if it was living or dead, they both produced tubes in response to the bacterium. So one of the things that we have looked at more recently is to ask, how in the world does this bacterium sense where it is located? Am I in an insect? Am I in a human circulatory system? And in order to answer this question and to understand how this bacterium is changing address and how it deals with the stress. Now, if you move from your apartment to a house, or from your house to the dorm, you know it's stressful. Imagine what it's like for a bacterium to go from a cozy little circulatory system to an insect, or vice versa. This is a stressful event, every bit as stressful as what we experience. And before we describe some of the data here, I just want to just go back in time and talk a little bit about some general biology. Make sure we're all on the same page. So we have DNA, the master hard drive for, or template for genetic information. at least for you and I and sandflies and bacteria. That is then transcribed into a messenger RNA, which then is translated to become protein. Now, some of the sneaky viruses out there can flip the coin and go backwards and do all kinds of things like HIV, but that's irrelevant at this point. Now, Mother Nature has been able to design several points where she can regulate this flow of information. You can regulate transcription, you can regulate translation, heck, you can even regulate the proteins at the very end by other proteins and cofactors and so forth. So one of the things that we did was to look at a type of regulation that's very useful for when you change your address very quickly. There's not a lot of time to switch gears. Boom, you're in the circulatory system of a host. And from other bacterial studies, we know that another RNA molecule called small RNAs, and they're called this because they're small, so they're only about 50 to 400 bases long. And what they do is quite remarkable. Bacteria will make them, and they bind to mRNA, and they can do several processes. They can make them more stable. They can help them degrade. Sometimes they'll do the opposite things on different RNA species, so that their role is quite remarkable. But the incredible thing is that they are fast forms of regulation. So when we change address as a bacterium, we can regulate production of the protein. And I've got a question mark there because sometimes it goes up, sometimes it goes down. But bottom line here is that we can coordinate virulence, metabolism. Can you imagine how the metabolism would change for a bacterium in blood versus in an insect? That's night and day. And stress. You know, how does it deal with our immune pressures and all of those things that we bring to bear to fight the bacterium? So what we did, and this was some of Sean Wachter's research when he was doing his Ph.D. in my lab a couple years back, and we decided we would simulate all the conditions that this little bacterium would experience, if I can be anthropomorphic. All right? So he's going to experience sandfly temperatures, human body temperatures, our blood gas, on and on, even his favorite cell types to invade, endothelial cells and red blood cells. So it's a lot of work under a lot of conditions. And then we asked, what are the small RNAs that are produced in response to these? And we found that there are over 160 species of these small RNAs that are produced. And we haven't the foggiest notion what they're doing or how they're regulating, so we can mine this. 7 of them are highly conserved across all bacteria. So we immediately say, yawn, throw those out, we don't care. But 153 of these are novel, and we can look at those and figure out what they are doing. And we can ask computers using software to figure out what are some potential targets here in the context of that genome. And we found that 24 of those small RNAs are human infection specific. So they like to be expressed in warm conditions with human blood gas, maybe human red blood cells or human cells. 8 of them are sandfly specific. And the computer can tell you, well, the sandfly specific, we've got some that are involved in making new amino acids. So that's cool. So its metabolism is shifting gears. In humans, here's one, the catalytic activity. So the enzymes are different in the human than they would be in a sandfly. So it tells us a lot about what is going on. And we can ask computers, in this case, we're looking at a sandfly-specific small RNA, so one of those 160 small RNAs called BBSR9. And we can ask, what are targets? And then we can make RNAs and say, do these 2 RNAs, the mRNA and the small RNA, do they talk to one another and change migration in a gel? So here is 1, 2, 3 that actually bind and talk to each other. And here are 3 more despite what the computer said, that are duds. They don't interact at all. So now we're trying busily to try and figure out, well, what in the world are these mRNAs doing, and how is the small RNA regulating them? Just one example, the FTSH mRNA encodes a protein, an enzyme that is essential in E. coli. So maybe it's essential in Bartonella bacilliformis. We don't know. So why are these genes important in a sandfly? And the human infection-specific sRNAs, we're just starting that work, and we've got a lot of things to do in the laboratory. So just to kind of tie this up, this is a very highly virulent pathogen, and we still don't know a lot about it, but we do know just from research in my lab and many others that there are several factors that help this bug do what it does. And we're closer to a vaccine, but Carrion's disease is still a neglected tropical disease. Until we can get some money from Colombia, Ecuador, Peru, where they've got to deal with this, or else it's just going to continue to kill people and afflict children and pregnant women and so forth. It's tragic. We still know very little about the epidemiology. That is, do we have additional reservoirs? Is it transmitted by alternate sandflies? And so forth. And we're going to look at those small RNAs that I just mentioned in the last slide and try and figure out how and who they talk to amongst the RNAs. Maybe we could find a drug target to stop the infection. So for some suggestions for ways of knowing our CLS seminar series this year, at least in terms of medical microbiology, I think it's really important to include patients struggling with the disease. Get out there, don't just look at the bug in your lab like a lot of my friends do. Get out there and see what it looks like to these patients and how they are struggling. Get out there in the field and capture sandflies. Look at how the disease is being transmitted. And that ties into this vector of transmission. And dissect that pathogen at both the molecular and cellular levels to figure out how it is making a living and causing this suffering. And then hopefully we can put all these together in a holistic fashion and come to a nice picture that can help us to eradicate this disease. So be happy to answer questions.
Thank you, Mike. That was as scary as it was interesting. I have just a couple things to tell you. When you speak, hold the microphone like this so we capture everything. And so we have our first question, so go ahead.
Yeah, it's really scary when you've been down to Peru and didn't know any about this stuff. But I'm interested in your first bullet right there. I'm sorry, include patients struggling with the disease. And I think this is more of an observation than anything else. I have a friend who's really suffering from long COVID, and they're really including those patients in that research. And it has just turned the medical community upside down because they don't have any blind studies. These people are already in it, and they're telling them. Is that a real problem, though, in terms of how you look at dealing with people that are already struggling with this? Is that new?
I think that bullet point there is harping on mainly on my colleagues. There are some microbiologists that tend to neglect and forget the patient because they're so interested in the pathogen and how it makes a living, the pathogenesis, without ever, so they might work with animal models, they might work with cell culture, like I showed you, but seeing the actual effects of the disease, the clinical end, is difficult for them. And granted, most of them are not physicians, so they don't have access or ready access to patients. Yeah. Yeah, Eleni.
if I can, I just want to remind the folks who are Zooming, if you have a question or comment, go to your Zoom chat function and submit your questions or comments through chat, and then we capture that. We'll be able to ask those questions. So, go ahead.
I thought it was really cool. I was wondering about how I noticed that you said that there's 2 phases of this infection, and each phase is treated with different antibiotics. That's crazy. I just wanted to know. I wanted to know what was going on with that.
Yeah, good question. Back in the older days, the locals could use chloramphenicol because it's dirt cheap, but it has so many side effects that it's been replaced with more modern drugs. Verruga peruana, the skin ailment, is relatively more difficult to treat than the acute hematic phase, the Arroyo fever. And the drugs that we would use for the Arroyo fever just have no effect whatsoever on the chronic, the Verruga peruana, perhaps because it's walled off so well. I mean, that would be the lowest hanging fruit to explain that observation.
Mike, if we had a situation where some Peruvian rich uncle died and left you all the money that you needed, how much faster could the research for this on this particular pathogen proceed to a cure?
Jeff, I've been a poor scientist and I've been a rich scientist, and I can tell you the research goes a lot faster with money. I think that if money were put into this and we could have clinical trials in the endemic areas and endemic zones, this disease could be eradicated. I'm certain of it. It's just a matter of finding the dollars.
Is there any hope from, say, the World Health Organization or any of the international groups?
Well, believe it or not, you would think so, because it's a neglected tropical disease. But if anything, it is at the bottom of it would be the neglected tropical disease. So they're focusing on bigger problems than Arroyo fever. So they're thinking more globally. For diseases such as polio, they're trying to eradicate polio. They're looking at getting rid of, some other examples might be yaws, which is caused by one of the serovariants of the organism that causes syphilis. So they're thinking more globally, whereas this is more geographically restricted, and it's just not quite as sexy to the Gates Foundation. World Health Organization, to my knowledge, doesn't necessarily have grant funding. So most of our dollars have come from the NIH and NSF, and I neglected to actually show you. I apologize to the NIH, my goodness, and all these past collaborators and graduate students here. So most of our money has come from the NIH. But even in this context, many of them have never heard of Bartonella bacilliformis, professional microbiologists. So it's truly neglected.
This was kind of based off of Eleni's. For the skin blood blister, is like any like topical or ointment effective at all, or is that ever going to be a possibility for treatment?
Not to my knowledge. There's nothing that would absorb in there and stop the infection. It has to be an antimicrobial that's given orally. Yep, or intravenously.
I've got your students here quizzing you.
So I remember you saying that the second phase of the infection, they just have chronic like septicemia.
Mm-hmm.
How do they live with that? Like, I thought that septicemia was like something that became fatal.
That's a great question. So the acute hematic phase, the Arroyo fever that killed 6,000 railroad workers, kills travelers, kills the unborn children, pregnant women. These are individuals that have not had repeated exposure to the bacterium. Whereas the children that we would see in Cosma, Peru, or in the Andes had grown up with this, and maybe they had a subclinical dose of the agent, and they slowly built up enough immunity to the point of reaching that equilibrium or carrier state, as we call it. So that is the simple answer to your question. But travelers that go in are exceptionally at high risk in the Andes region, and if they got a Roya fever, it could be very serious. And there are reports in the literature of this. But the endemic areas, those people they just grow with it and live with it. Yeah.
I was curious, do our friends down in the Bitterroot at the Rocky Mountain Lab do any crossover work that touches on this?
Good question. I have collaborated with folks at Dr. J. Carroll at Rocky Mountain Labs. He and I worked on Bartonella quintana, which is an agent of trench fever in our inner city homeless populations and can wreak havoc with them with respect to heart disease. But on Bartonella bacilliformis, no, none of them have collaborated to date. But elsewhere, different areas, England, folks in University of South Florida, and UC San Francisco have collaborated with me, and so forth.
Actually, I have a question too. Let's say one of these fine Montanans goes to Peru and picks up this fever, and beans is how there's a bacterial component to it. They come back to Montana. How likely are they to pass it along to other close contacts if they don't happen to have a little sandfly hitchhiking its way home with them?
Well, I can tell you, Leutzemia vericarum is one wimpy little sandfly. I'll tell you what, he'll die in your laboratory. He's just and he's very picky. But without regards to that, the chances are very, very slim because you would have to have blood-borne transmission to the next individual. And we've never even had a close call in the lab, for instance, where we're working with it and purifying it. Of course, we do it in a safety cabinet, and so we don't have aerosolization. But it would be virtually impossible unless that person gave a blood transfusion. Now, that is in the literature where patients had an unbeknownst infection with this agent, and they've given a transfusion and passed it that way. Yeah.
Since you showed that photo of yourself having been bitten several times by the sandflies,
Yes.
how is it that you yourself did not develop the severe form of the disease? Was it an immune question or a luck of the draw? Or?
No, that pardon me, my grandchildren gave me their cold, so no, those were just uninfected insects. So I allowed them to bite so I could just show you, the audience. That's the kind of person I am. I wanted you to see that. I wanted you to see what the insect bites look like, and they can really pack a wallop and give you a welt. But those insects were not exposed to the agent at all. No, no, no. Because these are Leutzemia longipalpus, which we get from Walter Reed Army Institute of Research. So they have sandfly colonies, again, your tax dollars at work, and they send out the pots of larvae, and then we release those and we use the adults. So they're clean in terms of that bacterium. God only knows what they have in that rabbit feces mix, but that's
so they
another
weren't
thing.
bites from your trip to Peru?
No, they were not.
Okay.
No.
This was just in the spirit of Daniel Carrion.
That's right, in the spirit, rather.
Not the actual practice.
Yes.
Are there any other questions? Here's one.
I can just.
No, I got the capture.
So you mentioned that this bacteria was transferred between maternal and the fetus. does that incur high rates of miscarriage just because of the developmental period and developing like the blood arteries and veins, how this can alter that?
Yeah, this is a real serious illness when you get vertical transfer from mother to offspring. It's not as severe as, say, syphilis, congenital syphilis, but it can be life-threatening to the baby, and for that matter, to the mother if she can't have some way to deliver that baby that's infected, or, it can be life-threatening, yes.
We have a Zoom question.
What would be the best way for travelers to protect themselves if they're in the affected area during peak transmission time, when the sandflies are more aggressive and potentially more likely to bite through normal bug spray?
I think prudence would be the best way, so using an insecticide. And for whatever reason, sandflies, although I think they're cute, they're sneaky little turkeys. They will bite you right around here, and they do so right around when the sun is setting. So avoiding that time period, insecticides, long pants. I know it can be pretty warm down in Peru, but those would probably be the best ways to avoid them. And then prudence in terms of after your trip, if you came home and you were spiking a fever and you felt really bad and you were starting to look pale, I think it would be time to check in and see if you have an infection, because it can be treated very successfully.
Any other questions? Oh.
The chick that asked that question, she wanted to know, do they respond to normal bug spray, or is it some specialized?
Yeah, a good DEET insect repellent would work excellent. Yeah.
Great. Well, I'm not planning to go to Peru, but I'm glad to know that you're on the job. For all future Peruvians and travelers to Peru, this was fascinating, and it's just one small slice of the kind of research that's being done on this campus today. We have a lot of amazing people at work doing wonderful things. So thank you very much. Another round of applause. And I want you all to make sure you return next week for our next lecturer. Sean Hill is a poet and is going to take us in a whole different direction on what another way of knowing, and I'm really looking forward to it. You know, for years we started the lectures with a poem. We didn't do that this year. We put a big weight on Sean. He's going to carry us next week for that. So thank you all for appearing. Thank you for those to you at home who watched through Zoom. And we'll look forward to seeing you next Tuesday at 7 o'clock. Thank you.